Hi class, good morning. So today we'll be learning about Mendelian inheritance. So let's go ahead, just jump right in. So have you ever wondered to yourself what determines your hair color? Or maybe the color of the flowers? Or why some people can roll their tongue and others can't? How many people in here can roll their tongues? Yeah, I can't. That's about as good as mine gets. It gets folded over. So there was a gentleman named Gregor Mendel who had similar questions and decided pea plants would be an easy way to experiment. He spent the next 7 years of his life cross-pollinating the various pea plants and recording the offspring's traits. So let's look at his experiment a little more in depth. He started with only purebred plants that he knew produced the same traits. So only tall plants produced only tall offspring, short plants produced only short offspring, and so on. Mendel also decided to observe discrete traits, meaning it was either or. There was no mixing of them, so it's purple or white flowers, wrinkly or smooth seeds, round or oval seeds, etc. He decided to study 7 different traits. For the purposes of today, I'm going to focus on flower color, just to make it a little easier. So, Mendel found when cross-pollinating purebred white-flowered plants with purebred purple-flowered plants that they produced all purple-flowered offspring. He then cross-pollinated those purple-flowered offspring with themselves and found that 3/4 were purple-flowered and 1/4 were white. Now, at the time, biologists thought the physical traits of offspring were a blending of the 2 parental traits. However, Mendel disagreed, having observed the first generation of pea plants having all purple flowers. If the biologists at the time were right about these certain traits, Mendel would have seen light purple flowers or a flower somewhere on the spectrum between white and purple, not distinctly white and purple. Mendel also observed that a phenotype not exhibited in one generation could reappear in the next. This led Mendel to believe that there were inheritable units, known today as alleles, and that the expression of one overpowered the other in certain traits. Now, today we understand that each trait Mendel was observing was controlled by a single location in our DNA, also known as a gene, and together all these genes make up our genetic composition, which we know as our genotype. And the differences in the expressions, physical expressions of these genotypes is called the phenotype. And these are determined by the alleles that we already talked about. And for genes expressing one trait or the other, like the traits Mendel observed, one allele is going to be dominant or expressed, and the other is going to be recessive or not expressed. So in the case with the flowers, the purple is dominant and the white is recessive. Now, if an organism has 2 of the same alleles, they're considered homozygotic or a purebred, such as the ones Mendel started with. But if an organism has 2 different alleles, they're considered heterozygotic or a hybrid. And a heterozygote is also considered a carrier because they have that recessive allele even though they don't exhibit that phenotype, and can carry it on to their offspring, where depending on the other parental genetic composition, that offspring may then exhibit that recessive trait. So a good way to show all this is through a Punnett square. In 1907, a man named Reginald C. Punnett devised a diagram to help predict the genotypes of a genetic cross. Today, we still use these as a way to help predict the probability that a certain offspring will have certain genotypes. And the way we denote this is through capital and lowercase letters. For each gene, we always assign a letter, so that way we can keep track, especially if we're using multiple genes. You know, I always think of the letter as like a variable. You assign your variable to your gene, and there you go. For today, I'm going to use A's. So I'm going to assign big A to my purple flowers and little a to my white flowers. And here you can see these are purebred because their alleles are both big A and the alleles are both little a, so they're homozygotic. Now when we cross them, we see that we get all hybrid offspring. Because they're heterozygotic, you can see they each have a big allele and a little allele. Now, what happens when we cross those with each other? So here we've got the big A, little a, big A, little a. So here we've got one homozygotic dominant because there are two matching alleles. Here we've got two more hybrids or heterozygotic dominant because they will produce those purple flowers. And we've got one white flower producing. So, as I've been saying, you can see all in the first generation will produce purple flowers because they have that dominant allele. And then in the next generation, we see that 3/4 will produce the purple and 1/4 will produce the white, um, such as Mendel observed. So really, what's his legacy? Before him, it was believed that phenotypes are only a blend of your mother and father, but Mendel showed that some traits are inherited as one or the other and not as a mix. Today, we understand this only works for traits controlled by one gene or by one locus in our DNA. Phenotypes like eye color, hair color, and height are a bit more complicated because they're controlled by more than one gene. So Punnett squares and crossing don't quite work out like that. But Mendel's efforts really changed our understanding of genes and genetic composition. Before him, we had no thoughts of genes or alleles or inheritable units like that. So he really just changed, he was really the foundation for modern day genetics. And for this last slide, I just wrote up some key terms for you guys just in case you needed some help studying. So I hope you have a great rest of your day.